This is one most important topic regarding the instrumentation of HPLC. In the previous sessions, come across the definition of HPLC and the principle of HPLC also. Yes, here in this session, let's see about the instrumentation of HPLC. Right, the first part, that is the solvent reservoir. The solvent reservoir is usually made up of high quality borosilicate glass. It should hold the mobile phase, and that should be innate, and it shouldn't release any of its contents to the mobile phase. An inline tube is there in the middle portion of the solvent reservoir, right? An inline tube we can see in the middle portion of the solvent reservoir. If the solute we are using a volatile, that inline tube should be covered definitely with a Teflon tape, otherwise the volatile solvents will evaporate. So to lose that, to avoid that tendency of the volatility of volatile solvents, we should cover with a Teflon tube, right? To the bottom of the inline tube there is a degassing module. We can see around 10 micron size, right? 10 micron particulate size on the degassing filter, right? It traps any particulate matter including gases also. Yes, if we use the buffers, if using water, so for the buffers preparation we use water will develop itself some sort of microbial growth. Yes, in that case, as a microbial growth will seen on the degassing module, it should be cleaned. For that reason it should be cleaned properly, otherwise microbial growth will seen. Yes, let's see what are the mobile phases that are used in the HPLC. Right, mostly HPLC grade solvents are used, and solvents after the degassing under the sonication only we should use as a mobile phase in this mobile phase reservoirs, right? We, in case of some sort of buffers are also used, and for the buffers preparation mostly HPLC grade water is used, right? After the preparation of this buffer, the buffer should be filtered under the 0.45 micron nylon filter and 0.22 micron nylon filters, right? Even if any other traces of the gas is present in the buffer that is prepared, that should be sonicated. And even after the sonication also, if any sort of bubbles are present, that is removed with the degassing module. This is the importance of the degassing modules. Degassing module will remove the dissolved oxygen and nitrogen also. Yes, it is not removed by these degasser. If it is not removed by the degasser, will leads to the pressure difference in the pumps, and the column response may vary, right? It leads to the detector response may last. This is about degassing module. Some sort of degassing modules are seen, namely vacuum filtration, helium spudging, sonication, and the online degassing module. Coming to the vacuum filtration, this uses the nylon filter for the filtration of the solid particulate matter. It is not efficient for the gas removal, right? Next one is the helium spudging. Yes, it has its own limitation. It is cost effective but is efficient. Placed inside the mobile phase reservoir. Because of its cost effective, it has the limitation that it is placed inside the reservoir, right? Afterwards, this is sonicator. Here is the, it looks like sonicator looks like this. It has the switch. This is the mobile phase. In this, the water is placed inside this. If you switch on this sonicator, and this was some sort of vibrations are developed inside this water, and the mobile phase placed in this tray, and if any sort of bubbles, gases or inside this mobile phase, these gases come close to one another, tends to fuse and then escape upon the switch of this sonicator. Yes. Afterwards, next one is online degassing module. HPLC tubes contains the semi-permeable membrane while the mobile phase passes through the tubes, right, to the pumps. It traps any other gases present in it. Next part is the pumps. Second part is the pumps. The pumps delivers the mobile phase to the column. Some sort of pumps we can see. namely pneumatic pump, hydrolytic resistant pump, syringe pumps, and the reciprocating piston pump. Pneumatic pumps are widely used in the preparative HPLC. Coming to the hydrolytic resistant pump, these are not widely used. Coming to the syringe pumps, it is seen mostly in GC, gas chromatography. Right, here in the gas chromatography, some sort of microliters is taken by the syringe and injected onto the column. Coming to the reciprocating piston pump, usually pumps uses some sort of liquid, takes some sort of liquid with time, microliter per minute. Right, it uses some pressure also. Let's see. It requires some flow rate and some sort of pressure is necessary to pump any sort of liquid. Right. Here is the reciprocating piston pump. Looks like this. Here is the two seals that we see, and a motor driven cam is also there. This is the piston upon the piston controls under the motor driven cam. Yes, if the piston moves to the left side, inlet check valve opens and the solvent reservoir enters to the pump. Right, upon the piston moves to the right side, solvent pushed up to the injector. Right, this is about the pumping. And disadvantage here is the flow fluctuation is seen due to the single piston is used here. Right, dual head reciprocating piston pumps are widely used. Here the excess solvent stored at the pulse dampers and that excess liquid sent to the next round. This is the use of the dual head reciprocating piston pumps. Next, what is the mechanism under which mechanism the solvent goes? Means here are the two mechanisms we can see, that is namely isocratic mechanism and the gradient mechanism. Here in the isocratic mechanism, single port through which mobile phase of one or mixture of solvents of constant composition we sent, means polarity remains, but the polarity remains the same here. Yes, here in the isocratic mode, in this single port through which the mobile phase of one or more solvents of constant composition can sent, but the polarity remains the same. Yes, here is the peak width is increasing and the time consuming process and poor resolution of the initial eluting peaks here seen in isocratic mode. Yes, high isocratic mode, look over here. Here are the four mobile phases and different composition, but the polarity remains the same for these mobile phases. And through the valve it goes to the mixing chamber and again goes to the pump, to the injector and to the column, and can goes to the detector in this isocratic mode. But in case of the gradient mechanism, these reservoirs of different polarities goes to the pumps with the different ports, namely binary ports are the quaternary ports are, look over here, binary and the quaternary ports we can see in the gradient mechanism. Right, namely what is next, we have question that is what is the gradient mechanism we see here. Here is the 20% methanol and 80% AC and acetonitrile at the 5 minutes time interval. After that 20 minutes we see 80% methanol and 20% AC and acetonitrile, that is seen after the 20 minutes. This is the gradient mechanism. As the time goes, the mobile phase composition is changes at the regular time intervals through the different ports, binary ports and the quaternary ports is the gradient mechanism. Here, two or more solvent goes to the mixing chamber under pressure used here are more because ports are more. Since two pumps are used for this, pressure used is high, and the advantage of this is decrease in the retention time, elutes faster, there were decrease in the retention time, and the peak height is higher, which is important for the trace analysis, and this we already seen. Right, next one is injector. The next part is the injector. The injector used here are the 6-port rotary injection we said. Why it is means here are the 6 ports that has the capability to rotate. Right, so it is the 6-port rotary injector. Yes, this has the two positions, position A and position B. Here in the position B, it takes the sample solvent sample in this position mode, it takes the solvent and inject onto the column here in this position B. Yes, in this position, let's see, here are the ports, that is 1 2 3 4 5 6, right? From the vial it takes a sample and that goes to the position 2, and this position 2 is connected through a loop to the 5th position, and to the 6th position it goes to the excess solvent. If it takes excess solvent, it goes through the outlet here. Some sort of required amount of liquid it stores, right? 5th and 6th position stores the loop, stores the 5th and 6th positions, it stores the sample. Upon the rotation to the anti-clockwise direction, the 5th and 6th position goes to the column side here. The 5th and 6th positions here upon the rotation comes to the column position and the mobile phase position, pump position, so that it is injected onto the column. Right, mobile phase pump from the solvent mobile phase goes to the column here. This is the position B. Yes, upon the rotation, the solvent taking from the sample and the mobile phase comes to the column position in the rotation. This is about the 6-port rotary injection. Next, the next part is, that is said to be the analytical column. It is the heart of HPLC, where the actual separation takes place on this column. Yes, on which the stationary phase is packed, made up of stainless steel, usually seen, that ranges from 5 to 100 centimeters, usually 15 centimeters, or said to be the shorter columns, 25 centimeters said to be the longer column. Here, the particulate matter of the silica bed is of 2 to 10 micron size. Usually used particulate matter of the columns are 3 to 5 microns are used, decreasing the particle size of the silica bed, increasing the surface area, increasing the resolution, and increasing the efficiency. That is used, mostly widely used one is 3 to 5 micron packing bed of the silica is most widely used. Here, by increasing the life of the column, guard columns or the sacrificial column is used here. In general, the stationary phase is in polar in normal phase HPLC, but in the RP HPLC, stationary phase is said to be the non-polar. How? That stationary phase in the HPLC, normal phase HPLC, has to be derivatized so that the polaric nature of the silica gets changed to the non-polar nature, right? After the chlorination, SiO is the siloxane group, HCl gas is removed here, and silica is attached here. This gets the non-polar nature. Most widely used columns are the C8 and the C18 columns are most widely used. Next, last one is the detector. Yes, this is said to be the brain of chromatographic system. It gives the perceptible impulse and can be interpreted by the data. Yes, there are some sort of detectors, namely bulk property detector and the solute property detector. Under the bulk property detector, refractive index and the dielectric constant detectors come under this bulk property detector. Yes, coming to the refractive index detector and the dielectric constant detectors, refractive index and the dielectric constant of the mobile phase is changed in the presence of an analyte, which detects the change in the intrinsic property of the mobile phase in presence of the analyte. From this, chromatogram is made. Yes, so coming to the solute property detectors, namely UV detector, fluorescent detector, electrochemical detector, and the miscellaneous detector. Again, in the UV detector, we see different types: fixed wavelength detector, variable wavelength detector, and photodiode array detector. Coming to the fixed wavelength detector, here a fixed wavelength is fixed in this fixed wavelength detector. Yes, mobile phase from the column is passed through a flow cell at fixed wavelength, fixed nanometers, compound elutes, and the flow cell held in the radiation beam of the UV spectrophotometer at fixed wavelength, it detects the compound. Right, this is about the fixed wavelength detector. Variable wavelength detector, it is widely used for the alkene, aromatic, and the compounds having the double bonds. This is useful. Yes, coming to the PDA detector, this is the most widely used one. When a polychromatic light is beamed onto the sample cell. It hits the sample. After the hitting the monochromatic light to the sample, on the other side, a monochromatic plate is placed at each and every wavelength, that is 200 to 400 nanometers of the UV range. At each and every wavelength, it detects the nature of the compounds, so that it gives the diluent peak, solvent peak, and the compound peak itself. It gives the 3D plots, so that is it is widely used one in the UV detectors. It has that much of importance in the detection. It gives the complete profile of the drug. Yes, coming to the fluorescent detector, the name itself indicating that fluorescent means emission, some sort of light is emitted. It come under emission spectroscopy. When a light hits the molecule, some sort of energy is absorbed by the molecule. That molecule goes to the excited state. The molecule in the excited state no longer remains. It has to come to the ground state by the emission, by the loss of certain energy. Thus, emission is said to be the fluorescent. Yes, that is recorded by the detector as the compound that is present. That the compound we are going to analyze by the fluorescence at certain wavelength. The light that is used here is the xenon lamp. That is about the complete description of the instrumentation of the HPLC. Subscribe my channel. For any queries you have, you can comment me also. If any topic you are in doubt, you should comment me. Don't forget about my subscription. Okay, bye.